Hi everyone, I'm Mohammed, a PhD neuroscience student in the addiction research group under Dr. Stephen Laviolette, and this talk will be about two of my projects, one that is currently ongoing, the title of this presentation, and they're both exploring how prenatal THC exposure induces a schizophrenia-like phenotype, alterations to cognition, anxiety, and emotional processing, and how a novel treatment can recover the deficits that are imposed by this prenatal exposure. The frequency of self-reported cannabis use amongst pregnant women is increasing year over year. And in fact, Young Wolf et al. Found that particularly over the past year during the pandemic, the number of pregnant women using cannabis has dramatically increased. As cannabis legalization occurs in much of the Western world, misconceptions about its fetal effects and its general use before pregnancy by the of-age populations has made studying the effects of prenatal cannabis all the more urgent and necessary. Unfortunately, exposure to particularly THC during gestation increases risk for childhood and later life psychopathology, with symptomology ranging from attention deficit hyperactivity disorder, increased anxiety, depression, and psychotic-like experiences. While the mechanisms remain elusive, it is well established that THC disrupts the endocannabinoid system. The system emerges during prenatal brain development, and its cannabinoid receptor 1 is critical to actually establish our neural circuitry and synaptic physiology, while its cannabinoid receptor 2 modulates microglia and in turn neuroinflammation. However, the role of the endocannabinoid system begins to shift by adolescence. It's one that is involved in the pruning of these same synapses it once helped form, and by adulthood, its role is in one of cognitive and affective processing. Importantly, THC disrupts both receptors by overwhelming them, which, when they're involved in early neurodevelopment, likely then leads to issues with synaptic physiology, viability, and the function of neurons throughout the brain. These processes are highly controlled, and THC is disrupting these fundamental processes. Of course, this suggests to us exposure to THC prenatally would be fundamentally more detrimental than later life usage. The double hit hypothesis of schizophrenia that suggests that prenatal THC delivers the first hit, causing an endocannabinoid imbalance in the fetal brain, establishing a vulnerable neuronal network such that a second hit would precipitate the emergence of a schizophrenia-like phenotype. However, this hypothesis was born out of the idea that an insufficient or subthreshold dose would actually reach the fetus, which in turn spurred on research that used such a subdose, a 2 milligram per kilogram dosage. But this 2 milligram per kilogram model only models essentially a joint, if you will, with only 5% THC content. The standard today is closing in at 10 to 25%. Recently, a new model for prenatal THC out of a collaboration with our group and Dr. Dan Hardy modeled 7 to 8%, a more ecologically valid model. That we set up that rather exploratory experiment with the hypothesis that a sufficiently high and ecologically valid dosage would induce long-lasting neurobehavioral abnormalities, a schizophrenic phenotype, without the need for further hits or exposure to THC. So we exposed dams to 3 milligrams per kilogram THC or vehicle saline from the first trimester until birth, gestation on day 7 until 22, and then explored the consequent phenotype. We first assessed anxiety, which is typically elevated in adolescent THC exposure models, as well as schizophrenia. The EPM and light dark box revealed that males are more anxious than females, are experiencing a greater anxiogenic effect, while the females were more resilient to that phenotype. Similarly, for social anxiety and motivation, we are again seeing that trend carry over. But importantly, prenatal THC is not reducing locomotion at any time point in the open field test, so alterations to locomotion do not account for that anxiogenic effect. These alterations in anxiety are likely to arise from a disrupted mesolimbic system, which is involved in anxiety and extends from the ventral tegmental area to subcortical limbic regions such as the nucleus accumbens. We assessed basal neuron firing at the VTA and discovered that males exhibit a hyperactive dopamine neuron firing coinciding with their enhanced anxiety, while females again appear to be resilient to this phenotype. For the cognition and memory tests, the results revealed that both male and female rats exhibit disruptions, as assessed by the social recognition test, the longer-term recognition memory, and of course, working memory. These disruptions, while possibly disrupted by the mesolimbic issues, are actually governed by mesocortical circuitry, with the two major components being the prefrontal cortex, the PFC, and the hippocampus. Surprisingly, the female PFC and hippocampus showed greater glutamate neurons firing, while the male hippocampus showed a hypofunction. We then further analyzed oscillatory activity, the communication and synchronization between neurons in the PFC and hippocampus that is actually known to underlie cognition. A stark sex-dissociable difference was also observed here. Clearly, the male THC and vehicle oscillations in the ventral hippocampus appear almost completely decoupled. The male hippocampus has decreases in theta, alpha, beta, and gamma, and an increase in the delta oscillations, with the female PFC exhibiting significant increases in gamma and a significant decrease in the hippocampal delta. All of these suggest disrupted cognition, but again, the female progeny are apparently more resilient. But why is any of this happening? What underlies these changes? Well, let's go back to the endocannabinoid system. This right here is the endocannabinoid system in its entirety, as suggested by Villo and colleagues in 2019. But I want to draw your focus onto fatty acids. These make up a considerable amount of the system, both as critical molecules in the structure of the membrane. All of this is housed in the synapse, or once released from the membrane, they can also act as signaling molecules. The synaptic membranes are actually composed primarily of two major polyunsaturated fatty acids, docosahexaenoic acid, DHA, and arachidonic acid. Interestingly enough, both are actually critical to the production of endocannabinoid system agonists that allow for the broad roles that the system has with neurodevelopment and laying the groundwork for the synapses. Arachidonic acid can produce the two major endocannabinoid agonists, anandamide and 2-AG, while DHA produces an agonist called docosahexaenoyl ethylamine, DHEA, which, while weaker in affinity than the two major ones, is present at several hundred times the concentrations. So I want to be clear here. Before production into these agonists, they interact with the endocannabinoid system, and after production into these agonists, they're critical for it to work. But why is this important? Interestingly, it's also long reported through postmortem analyses that schizophrenic patients actually have significant reductions in membrane-bound polyunsaturated fatty acids, particularly in the cortical regions of the brain, like the PFC and the hippocampus, as well as some mesolimbic structures like the nucleus accumbens. But these membrane deficits are thought to be due to neurodegeneration by the authors that consider schizophrenia to be neurodegenerative. So to them, fatty acids are released from the membrane during the early stages of schizophrenia. But could it be more fundamental in its basis in that the deficits are actually present much, much earlier? We can't really answer that question simply relying on post-mortem analyses, but to shed some light on that, what would happen if we were to limit DHA during gestation and early development? DHA is an essential fatty acid acquired through the maternal diet by the fetus early in life. Well, the Cohen-Corey study showed that maternal dietary deficits in DHA cause molecular changes in the offspring's developing brain that result in the constrained growth of neurons and the synapses that connect them. Rodent models have actually then associated this dietary deficit with enhancement in anxiety and poor working memory, but we don't see issues with sensory integration and other forms of memory typically seen in schizophrenia. So of course, DHA is not the whole story, but the similarity of the molecular and behavioral phenotype does raise a really good question. Does THC interact with and disrupt the fatty acid profile during the period the animal is acquiring it? To explore that, we next conducted matrix-assisted laser desorption ionization mass spectrometry imaging. And that's just a fancy way of essentially saying that we looked at the cortical membrane-bound fatty acid profile to assess how prenatal cannabis exposure may be interacting with the fatty acid profile. It turns out that there is indeed a sex-specific phenotype here. The males are exhibiting significant reductions of cortical membrane-bound DHA, with the females again exhibiting a resilient phenotype. We then wanted to assess whether this deficit was actually present early on, and it wasn't due to some later life predisposition, that it was indeed neurodevelopmental. So we assessed PND21, prepubescent childhood brains. For the males at 3 weeks old, it seems like the results are kind of consistent with adulthood in that the fatty acid reductions at PND21 endure into PND120, into adulthood. But for the females, I think more critically here, contrary to the resiliency we've been seeing until now, behaviorally, electrophysiologically, the female progeny are exhibiting broad, significant reductions of cortical DHA. All of this has recovered by adulthood. So we next wanted to actually characterize the synaptic membrane structure of the membrane and see whether or not it was also affected. DHA is actually present primarily within 3 membrane phospholipids, phosphatidylserines, choline, and ethylamines, PS, PE, and PC, respectively. DHA primarily is within PS, and PS is primarily within the membrane, synaptic membrane. We assessed all 3 to see whether prenatal THC disrupted the synaptic physiology on a fatty acid structural level. To our surprise, indeed, prenatal cannabis exposure did in both males and females, but much like the DHA deficits, the reductions were more profound at childhood. However, importantly, the deficits remain in the males into adulthood, while the females have largely recovered their membrane deficits, except for some reductions in the synaptic membrane. So what's going on here? Why are the females experiencing this recovery? Well, estrogen and DHA have actually long been known to interact. DHA is a critical molecule for neurodevelopment, so the mother typically stores more DHA than the male counterparts to provide DHA to their offspring. Estrogen is also a G protein coupled receptor, which requires DHA-rich lipid rafts housed in the membrane to function, but these estrogen receptors actually act synergistically with DHA and can favor the reuptake of more DHA into the synaptic membrane. And several groups have actually explored how estrogen and DHA production are linked. When the female or male rat meet DHA, they can activate several different potential molecular machinery to enhance production. Of course, female rats can get a lot more out of this since they have a lot more cycling estrogen after puberty. Perhaps then THC is preventing the appropriate levels of fatty acid accumulation in the brain, thereby mimicking a deficit seen in the dietary deficient models. Once female rats hit puberty, they're able to recover and deviate from the male PCE fatty acid phenotype, normalizing their phenotype. So female rats have an endogenous compensatory mechanism of fixing their cortical DHA content. Why not supplement it during the period of logarithmic growth to mimic these endogenous mechanisms? Past DHA nutrition studies suggest that maternal diet can significantly enhance cortical DHA content of their fetal offspring, and so this suggests a potential target for fatty acid amelioration in both male and female rats. Is a prevention of the prenatal cannabis-induced phenotype possible? Well, let's finally get into the prevention study. So we hypothesized that prenatal THC exposure induces schizophrenic lipid pathology, and so we predict that DHA supplementation may actually limit the behavioral and neural phenotype. Here's a quick overview. So we did the same 3 milligram per kilogram THC model as prior, only this time we manipulated the maternal diet, increasing the DHA content of their diet. The diet ends at PND21, parturition, weaning, up until which the progeny get DHA from lactation. We also wanted to try characterizing the phenotype during adolescence and adulthood and see whether or not there are some carryover effects from adolescence into adulthood. The data revealed that the omega-3 groups, which are these shaded groups right here, almost consistently are less anxious. We see this in the EPM and the light dark box, but more so in the males, again, of course. But more importantly, this benefit actually extends into adulthood. Looking at the EPM, the THC-N3 group is significantly less anxious in the males and females compared to the THC control group, which are these right here. So the omega-3 diet is preventing the anxiogenic effect produced by prenatal cannabis exposure in the males. But even though females don't really have that issue, looking at the control groups, the omega-3 groups are still a lot less anxious. We see that trend continue with social motivation as well, both in adolescence and, again, importantly, the benefits remain into adulthood. Also, the diet is not affecting locomotion, so that does not account for the deficits. For cognition, we see similar trends. Both the males and females are seeing a rescue, or better yet, a prevention of the THC-induced phenotype in adolescence for social memory, recognition memory, and working memory. We see this benefit actually extend into adulthood as well. The N3 diet is correcting the deficits where they appear. But how about electrophysiology? For adolescent neuron spiking activity of glutamate neurons in the PFC and hippocampus, we are again seeing that benefit from the omega-3 diet. So this is very promising, of course. But we then wanted to see if DHA was actually correcting the synaptic membrane deficits and fixing the issues. Like if that is indeed underlying everything, it should be fixing that issue. So here's a fatty acid analysis, a comparison between the 4 treatment groups. But I want to focus your attention to these ones right here. These are the control groups where THC has significant deficits compared to the vehicle controls and to the THC group given omega-3s, and the THC group given the omega-3s has a similar amount of DHA content as the vehicle control group. We see this in the male and female prelimbic and infralimbic regions of the PFC and in the nucleus accumbens. We see this also for the hippocampus, suggesting that the diet is indeed fixing, ameliorating the cortical synaptic membrane deficits in fatty acids, and seemingly also correcting the behavioral and electrophysiological phenotypes. Unfortunately, I haven't yet analyzed the adult electrophysiology or the MALDI just yet. It is an ongoing project, but just the behavioral improvements, the adolescence electrophysiology, and these childhood MALDI results suggest a very promising prevention in both males and females, with the very simple manipulation, the addition of primarily just DHA into the maternal diet. So the preliminary results reveal that interventions may be possible to ameliorate much of the deficits prenatal cannabis exposure induces. Of course, much more preclinical and of course clinical research is required before we can assert anything. But apart from this intervention, improvements in primary prevention of use and education is necessary to inform women of risks of using cannabis. The next steps for this project really is, of course, the analysis and publication of response data, and then to conduct a follow-up analysis to assess the critical window of supplementation that allowed for this rescue. Was it indeed the prenatal period, or could postnatal rescue also ameliorate the behavioral and much of physiological deficits? Well, I hope to see everyone here next year to update everyone. And with that, thank you so much for listening. I have, of course, several acknowledgements to make. Thank you to my colleagues, my advisor, Dr. Stephen Laviolette, and my community for their unending support. Thank you.
Hi, I'm Angelica Silva from Neuroimaging in Mental Illness Robert Research Institute at University of Western Ontario. Today, I want to share my presentation entitled Syntactic Complexity in the Diagnosis of Schizophrenia, a Belief Propagation Model. To begin with a disclosure, I declare no conflict of interest regarding the topic and content of this presentation. In terms of the agenda, it's based on 3 learning objectives. First, basically, we'll learn why the way to put the word together syntax is a key feature of human language. Second, we need to link this linguistic background in order to appraise the evidence for language abnormality in psychosis, case schizophrenia. And third, learn the utility of combining automatic natural language processing, NLP, and Bayesian network belief propagation model to increase the accuracy of clinical diagnosis. Moving on the first objective, we need to consider language as an essential source of information in psychiatry, including thought and other cognitive functions. For methodological purposes, language is divided in 5 discrete levels, and one of those is syntax. However, the syntax structure, therefore complexity, is not only a linguistic phenomenon. Why? Because when we talk, the hierarchy between no, the simple word we use day to day, increases dominant relations or type of connection. Therefore, the speaker oral production can be unlimited, showing us the richness of language. Please look at the graphic representation, the parse tree of a simple transitive clause. Smart women like smart men. Is telling you how the network of know is ranged, considering first higher level, for example, the word combination based on syntactic rule that give a word, for example, a type of grammatical role such as subject, and lower level which imply the way as phonemes combine into word. To put in context the importance of syntax in the architecture of language, I have to say that the study of syntax called for a property of complex syntax named recursivity. It characterizes the key feature that separate pre-human organisms, even human pre-language child language, from human language that we are discussing now. With this in mind, we also must answer the question, what does syntactic complexity mean? Note that the concept itself is indeed complex and refers to sentences that deviate from the basic canonical word order in English, subject, verb, object, to sentences with varying degrees of, for example, embedded clauses. At the top of the graphic representation, we can see how a simple clause, women are smart, is the result of the agreement between grammatical role and dependency relationship. However, in women who like smart men are smart, you can identify closest boundary, see all the words in blue. This graphic representation shows as a crucial differentiation between independent and subordinate clauses due to the linguistic process of embedding that use clauses inside other clauses. Then another important question shows up. Can any linguistic dependency be reduced? The answer, any question that could be reduced complex grammar with signal of poverty of speech expose a language abnormality. Second learning objective. I must say that work has been done to measure features of syntactic complexity correlated to clinical symptoms according to TLI and PAN assessment in clinical setting. This has been created the opportunity to look forward negative and positive language symptoms in patients in the schizophrenia spectrum, especially those named as traditional index, such as reduce the number of words, length of sentence, graded pausing. However, none of those words look for progressive changes in complex clause grammar, which is our main goal. The last learning objective is telling you that the analysis of linguistic features requires automatic technique as natural language process analytic to get objective measurements like the open source we use task, which explain later. And training language models such as Bayesian network to establish the causality on variable because of probabilistic reasoning. Now, please look at the image. Belief propagation is an algorithm to infer hidden probability. Name beliefs, the blue circles, within a network giving a hidden probability distribution in another distant node. I know it sounds weird. Let me explain a little bit. In the case of Bayesian network, the algorithm allowed to infer causality between nodes because of the generative model. For example, the hidden probability of schizophrenia propagates in terms of a message green line through the network until reaching the node corresponding to the observed symptom. For example, disorganized speech. By reversing the message passing red lines, we can infer the hidden probability of the pathology or diagnosis. Now, the current research where we apply the combined model respond three research questions. First, is longitudinal change in complex clause grammar associated with the consensus diagnosis? Second, what is the probability of increase or decrease in clause complexity after 6 months of initial assessment? Third, does a longitudinal change in clause complexity predict a consensus diagnosis? Then we set up the language data acquisition from a time baseline 0 month to 6 month follow-up approach. Our sample size was n equal 38 composed by 12 healthy control and 26 patients first episode psychosis at baseline. Then after having a psychiatric consensus, 18 out of 26 were diagnosed with schizophrenia and 8 with affective psychosis. All participants described 3 pictures, 1 minute each from the thematic appreciation test. 2 moments, baseline and follow-up. Finally, we record and transcribe 228 speech, and we use TAS, which is an open source for the automatic fine-grained analysis of syntactic complexity in order to get indices of clause complexity. Turning to the result, we can affirmatively answer all the result questions. Result 1. We fit a data-driven Bayesian network model, belief propagation, to 26 clause complexity index, group membership at time of first assessment, and group membership at consensus diagnosis time, which means 6 months later. The resulting model showed that one of the 26 clause complexity indices, nominal soldier per clause, please look at the red square at the top, was causally associated with the consensus diagnosis, even though the remaining 25 indices were mutually associated but independent from the diagnosis. I'm pretty sure that at this time you are asking what is a nominal subject per clause. It is a group of words that are like a noun in a sentence. Please look at both examples, the words in bold. Depending on the grammar, nominal subject per clause can be understood as a noun phrase. In this analysis, we consider noun phrase without pronoun, which is possible in English. This index was the average number of nominal subject per clause detected in the transcripts and divided by the total number of clauses. So, where does it take us? To interpret the patient's turn in reducing the complexity of grammar to simplify a structure of nominal subject per clauses, although in spoken language we expect construction with more information in this position, reflecting the referential processes, the process of describing object ideas, etc. Result 2. After identifying our index of clause complexity, we were able to estimate the probability of observing a longitudinal chain. Longitudinal chain was computed by subtracting the index value at baseline from the value at the time of diagnosis. The Bayesian network revealed that a healthy control subject, please look at the black bar on the left, had 100% probability of increased clause complexity in their speech. In contrast, patients with diagnosis of schizophrenia are highly likely 70% to show a decrease in clause complexity across time. Please see the tall red bar in the middle of the graph. A lower probability of decrease in clause complexity, the smaller red bar on the right, was estimated in patient diagnosis with affective psychosis. Result 3. It is the most interesting finding of this study. The association between the longitudinal chain in clause complexity and the probability of being diagnosed with schizophrenia. So far, the belief propagation model revealed that changes of receiving a diagnosis with schizophrenia increases when clause complexity has taken negative value, which means use a simplified noun phrase structure. See again in both examples the word involved and how the line is getting up when we are talking about negative value. Now, concluding remark. To sum up, schizophrenia causes a longitudinal change in the syntactic clause complexity of spoken language. The probability of being diagnosed with schizophrenia increases longitudinally when patients use fewer nominal subject per clause, conjoin lower clauses subordination dependency, which is not expected in formal speech. Both features comprise the bulk of morphosyntactic machinery of human language, deteriorating from the onset of the illness. The study of progressive chaining complex clause grammar based on the combination of NLP and Bayesian network might create the opportunity to increase the accuracy of diagnosis, prognosis, and perhaps monitoring relapse of symptoms of schizophrenia in clinical context. Then I have to say thank you for listening, and I'm glad to answer any question.
Hello everyone, my name is Charlotte. Welcome to my talk. Today I'd like to discuss with you on why and how I discovered two subgroups of patients with psychotic disorders based on cortical thickness. I will firstly briefly talk about cortical thickness in schizophrenia, how and why we should look at patient subtypes, and finally, how these two subgroups were similar and different from one another. To start off, cortical thickness is how thick the gray matter layer is in our brain. It is measured by the distance between the red boundary and the yellow boundary as shown in the picture here. The thickness of the cortex can reflect the amount and density of the neuron distribution. And for this reason, it is quite often studied in neurodevelopment, aging, and in different diseases like Alzheimer's and schizophrenia. By measuring this thickness, we can see the health status of our brain, and this feature is also found to be associated with cognition. So what does research tell us regarding the relationship between cortical thickness and schizophrenia? It actually shows that patients generally have a lower thickness profiles compared to the healthy populations, especially in some specific brain regions. Another finding is that this abnormality is observed across different stages of the illness, including clinical high-risk patient, recent onset patient, drug naive, as well as chronic patients. A lower thickness is also found to be associated with certain types of symptoms, symptom severity, cognitive performance, and treatment responsiveness. However, what we cannot conclude at this point is where and how some regions become thinner than the healthy levels, and the degree of thinning varies between studies. One explanation for this is that subtypes of psychosis exist. If we include patients with different cortical thickness patterns into the same group and study them, then it will be difficult for us to see the locations or the extent of cortical thinning. And the concept that there are patient subtypes in schizophrenia is actually not new, because patients with the same diagnosis can have very different clinical profiles and trajectories, have very different cognitive abilities and functional outcome. So it wouldn't be surprising for us to see that patients would have a vastly different neurobiology as well. And one way to solve this subtype puzzle is to use cluster analysis. So cluster analysis is a statistical procedure that can divide data objects into subgroups based on similarity. In my study, we collected data from 3 different cohorts of patients and healthy controls. And these 3 data sets were named Neurocognition Project, TOPSY, and CON project respectively. These patient groups are in different stages of the illness with different illness durations. In the NeuroCog project, it mainly included chronic medicated outpatient, and the TOPSY project included drug-naive first episode psychosis. And the CON project recruited patients in their stable phase of the illness. With these 3 datasets, we can run the same statistical procedures and see if their results would converge onto a consistent answer. And if they do, then we can conclude that what we found from one dataset can be applied for patients at different stages of the illness and for patients with different treatment status. Here, I'm showing the results that all 3 datasets converge into a 2-cluster solution. And in cluster 1, as can be seen in the bar plot here, cluster 1 mainly composed of patients with percentage ranging from 75%, 83%, and 100%. And we can see that cluster 2 has relatively evenly distributed patients and controls. On the right side here are heat maps of their cortical thickness patterns. Each row here represents a participant, and each column represents one cortical thickness value. The rows have been reordered, so participants in cluster 1 are on top. In terms of the color blue here means a lower thickness, and red represent thicker cortex. And just by looking at the heat maps, we can draw a horizontal line in each of the heat maps to separate the 2 clusters. And we can also see that cluster 1 generally have darker and larger areas of blue, which means that cluster 1 is the subgroup that has reduced cortical thickness patterns. When we look at other characteristics of the 2 subgroups, we found that there is no difference in terms of symptom severity, except for the CON dataset. Comparisons of functional outcome of the 2 subgroups tell us that the 2 subgroups did not differ. Antipsychotic exposure did not differ between the 2 subgroups either. Furthermore, we don't see a difference in terms of illness duration. So with all these negative findings, and we can say that this difference in cortical thickness patterns did not translate into clinical subtypes with different levels of symptom severities and functional outcomes. And then we further look at patients' cognitive performance. And since cognitive measurements are only available in the neurocognition dataset, when we compare between the 2 subgroups, what is very interesting here is that in the healthy population, cognitive abilities vary with cortical thickness. But with schizophrenia, patients with a relatively normal thickness patterns perform almost the same as the patients with reduced thickness. We further run some exploratory analysis, and when we look at the correlation between negative symptom severity and cognitive performance, it shows that cluster 1, so the subgroup with reduced thickness, tend to experience negative symptoms and cognitive impairment at the same time. But this is not the case for patients in cluster 2. And when we look at correlations of negative symptom severity and antipsychotics exposure, it shows that the correlations of these two variables differ between the two subgroups. This could mean that thickness plays a role in the negative symptom treatment responsiveness to antipsychotics. To sum up the findings in this study, we can say that there are two profiles of cortical thickness patterns in schizophrenia across different stages of illness and treatment exposure. And we can also see that around half of the patients had preserved thickness patterns that are very similar to the healthy population. Next, we can also say that cortical thinning does not necessarily reflect a progression to a more severe clinical status or a lower cognitive performance, but a lower thickness would affect co-expression of symptom domains and imply symptom persistence. And with this, that concludes my presentation. And in the end, I would like to thank all funding parties and thank everyone involved in the studies, especially my supervisors, Dr. Leniapin and Dr. Walter Harridge. Thank you all for listening in. I look forward to hearing your thoughts or questions.